Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like and subscribe the channel. In the last session, we studied about plasma membrane and membrane protein. In today's session, we will study tight junction. When two cells are in close approximation in certain areas, instead of 4 layers, only 3 layers of plasma membranes are seen. This tight junction permits calcium and other small molecules to pass through from one cell to another through narrow hydrophilic pores. Some sort of communication between cells thus results. In this diagram, we can see tight junction between subepithelial cells. Absence of tight junction is implicated in loss of contact inhibition in cancer cells. Tight junctions are also seal off subepithelial spaces of organs from the lumen. They contain specialized proteins such as occludin, claudins, and other adhesion molecules. In this diagram, we can see tight junction are bound by claudin and occludin. Coming to myelin sheath, it is made up of the membrane of Schwann cells condensed and spiraled many times around the central axon. The cytoplasm of Schwann cells is squeezed to one side of the cell. Myelin is composed of sphingomyelin, cholesterol, and cerebroside. In this diagram, we can see that myelin sheath is formed around axon. These cells are also called as Schwann cell. Myelin sheaths thin out in areas, certain regions known as nodes of Ranvier. Due to this arrangement, the propagation of nerve impulse is wave-like and the speed of propagation is also increased. In this diagram, we can see that axon contains node of Ranvier between two Schwann cells. Upon stimulation, there is rapid influx of sodium and calcium, and so that depolarization occurs. Voltage gradient is quickly regained by ion pumps. The ions flow in and out of membrane only where membrane is free of insulation from Schwann cells at nodes of Ranvier. Hence, the wave-like propagation of impulse occurs. In this diagram, we can see that entry of sodium from extracellular space to intracellular space is ATP dependent. In multiple sclerosis, demyelination occurs at discrete areas. Velocity of nerve impulse is reduced, and this is leading to motor and sensory deficits. In this diagram, we can see that in multiple sclerosis, demyelination occurs at Schwann cells. Before moving further, I want to tell you that please like, share and subscribe the channel. Your one click on the subscribe button shall motivate me to prepare further videos. Also, any suggestions can be given in the commentary box. Coming to the next, microvilli. Microvilli of intestinal epithelial cells and pseudopodia of macrophages are produced by membrane evagination. This is due to the fluid of the membranes. In this diagram, we can see microvilli formed by membrane evagination. Membrane or membranes of organelle. Membranes of endoplasmic reticulum, nucleus, lysosomes, and outer layer of mitochondria may be considered as variants of plasma membrane. Percentage of protein content varies from 20% in myelin sheath to over 70% in the inner membranes of mitochondria. In this diagram, we can see different layers of membranes of endoplasmic reticulum. Coming to cytoskeleton, human body is supported by the skeletal system. Similarly, the structure of a cell is maintained by the cytoskeleton present underneath the plasma membrane. The cytoskeleton is responsible for the shape of the cell, its motility and chromosomal movements during cell division. The cytoskeleton is made up of network of microtubules and microfilaments which contain the proteins spectrin and ankyrin. Tubules consist of polymers of tubulin. In this diagram, we can see that different contractile proteins present in the cell, for example, actin filaments, microtubules, and intermediate filaments. Coming to next, molecular motors. Proteins that are responsible for coordinated movements in tissues and cells are referred to as molecular motors. These may be ATP driven, as in the case of the contractile protein, actin, myosin in the muscle, as well as dynein and tubulin in cilia and flagella are examples of molecular motors. Kinesin is which is mediates movement of vesicles on microtubules also requires ATP. In this diagram, we can see that contractile proteins are ATP dependent. Here ends our session. Kindly like, share, subscribe the channel. Thank you.